Okay, we'll continue with the cell cycle. As you know that cell cycle consists of two basic phases. Cell cycle, the how cell reproduces, or cell divides, how cell parent cell gets converted into the daughter cells or progeny. And that is called cell cycle. It keeps on repeating. Cycle repeats. And because of the cell cycle, cell replicates. One cell becomes two, and two becomes four. A four becomes eight. The cell cycle, it consists of two basic phases: interphase and mitosis. A mitotic phase. Interphase and mitotic phase. The interphase is again divided into three substages or subphases: the G1 phase, synthesis phase, and G2 phase. Mitosis phase, which is also known as equational division because the number of chromosomes remains same, there's no decrease or increase in the number of chromosomes, is again subdivided into four phases: prophase, metaphase, anaphase, and telophase. This actually is the division of the nucleus, not the entire cell. It's only the division that is occurring inside the nucleus, and it is called karyokinesis. Not the division of the cytoplasm that is around the nucleus, but the division of nucleus is karyokinesis. The mitosis, it is karyokinesis that happens inside the nucleus of the cell. Prophase, metaphase, anaphase, and telophase are the four different subphases of mitosis. So what we are going to learn today is the first phase of mitosis, the equational division, prophase. Prophase is marked by the initiation of condensation of the chromosomal materials. So inside the nucleus, chromosomes are there, which contains our genetic material, the DNA. The DNA inside it is made up of DNA. Okay, those chromosomes, the chromosomes are before the start of the mitosis. Although the cell grows and the genetic the chromosomal materials it duplicates in the synthesis phase. So what we see in the prophase prophase is what it is coming after. After synthesis and the G2 phase comes the prophase. And after this again will come the interphase, right? So there is condensation in the beginning before the mitotic phase. The chromosomes, they are just like threads. They will appear like threads, just like this threads, right? So it will start because of duplication of the DNA materials. It will start thickening, or there will be condensation of the chromosomal materials. So what they become mitotic chromosomes, right? So these are the chromosomes. Actually, chromosomes are what they are actually are. They are composed of two chromatids. So each chromosome that we see, they're actually two, consisting of two chromatids, two threads together forming one chromosome, right? So they are not like chromosomes. They are not like separate single thread. They are actually combined or composed of two chromatids. Chromosomes are composed of two chromatids. For example, this is one chromatid. These are chromatid, and they are joined together at the centromere. Centromere is a part where two chromatids appear to join or condense together here. Right. Suppose this is a cell, and inside and this is a nucleus. What we see during the prophase is that the nuclear envelope, the nuclear membrane, it starts disappearing, and inside it we found that we were finding that centrosomes, like dot-like structure, their material, the quantity of centrosome has also increased during the interphase. It has become double during the interphase, especially in the synthesis phase. They also got double, just like the DNA materials. So there is 2 times the increase in the centrosome, and those centrosomes they started moving towards the poles. Suppose this is a globe, just like our Earth. Suppose cell is a globe, the nucleus is a globe. So they started moving towards the poles during the prophase. The centrosome becomes twice and they started moving towards the pole. Not only that, we find that there's a distinct protrusion, a small protrusion of two microtubules called asters from centrosome, right? So so when we watch a cell inside a microscope, we'll find that we'll find that the Golgi complexes, the organelles, right? The cell organ, the Golgi complexes, the endoplasmic reticulum, the nucleolus, and the nuclear envelope, they will start disappearing. There's disappearance of these organelles. When we watch under the microscope, it means that the prophase stage has started. Disappearing of this or cell organelles means the prophase stage in the cell cycle has started. There won't be any Golgi complexes, there won't be any endoplasmic reticulum, there will be disappearance of nucleolus, and there will be disappearance of nuclear envelope. Hope you understood.